Hi everybody. We are back today to begin talking about protostomes. So for the rest of the semester we'll be talking about different kinds of animals and moving through the various animal groups. And so right now today we're going to be talking about flatworms. So it lines up pretty well with lab because last week we talked about flatworms. So remember, in the bilaterian animals, those that are bilaterally symmetrical, you have two big groupings. The first are the protostomes, and that's who we're going to talk about today, and the deuterostomes. We are deuterostomes. And just remember, these groups of animals differ in four big ways, right? So just to review, the protostomes, you have differences in cleavage. So protostome embryos exhibit spiral cleavage, right, where cells are added in a spiral configuration, whereas deuterostomes exhibit radial cleavage, where cells are put right on or develop right on top of each other. All right. Secondly, they differ in the fate of those embryonic cells. In protostomes, you see determinate development very early, so that if you were to excise a cell from a developing embryo, it couldn't develop normally or develop at all. It would arrest. Whereas in the deuterostomes, you have indeterminate development. So if you were to excise a cell, you could still, in an early stage of development, you'd still have a normal embryo develop. The third is the fate of the blastopore. So remember, here you have your blastopore and a protostome embryo and a deuterostome embryo. So the blastopore in a protostome becomes the mouth. That's where they get their name, right? First mouth. And then the deuterostome's second mouth, the blastopore becomes the anus. And the fourth big way that protostomes and deuterostomes differ is how the phylum in the animal is formed. So in the deuterostomes, you have the pouching off of cells here that will eventually become the phylum. So they're growing out and then they'll pouch off, excuse me. And remember, this is mesoderm tissue. It's one of those three germ layers. And then in the protostomes, you have basically just cells growing apart from each other. They don't pouch off from here, okay, as they do in the deuterostomes. So those are the four big ways that protostomes and deuterostomes differ. It all has to do with embryonic stages, embryonic development. Okay, great. So there are two big clades. So you see here's this phylogeny so I told you we were going to get into that and I would explain it all later. Well, here we go. So within the protostomes, you have the spiralia and the ecdysozoa. So the ecdysozoa, this is named for the process of molting ecdysis. So these are molting animals. And then the spiralia, these all have spiral cleavage, but they're called spiralia because of the spiral cleavage. All right, so I just said it, but just like in class. Remember, so embryos develop using what kind of cleavage? Spiral, that's right. Okay, most of these organisms live in water. They're aquatic, okay, and they move using either contractions of their body musculature or cilia, okay. And within the spiralia, you have two groups. You have the platyzoans. This platyzoa means flat animals. So platyzoans and lophotrochozoans. And those animals are named for a feeding structure and the name of their larva. It's actually kind of mishmashed together, but we'll talk about that when we get to the lophotrochozoans. So you don't have to worry about that right now, because today we're just going to be talking about platyzoans, and particularly the Platyhelminthes. Okay, cool. So these guys, again, they get their name because platyzoan. So platy means flat, zoa is animal, right? So they were given this name to mean flat animals. And they're pretty simple. They do not have circulatory systems. They don't have respiratory systems. They don't really need them because they most of them, their bodies are so flat. I shouldn't say most of them, all of them. Their bodies are so flat that you actually have gas exchange right across the surface of the animal. So they don't have to worry so much about moving oxygen into their tissues and CO2 out because it diffuses because they're little flat buddies. Okay, but so even though they don't have these circulatory or respiratory systems, they do have complex reproductive systems that ensure the continuation of these animals. And what's interesting is we'll talk a little bit about their reproductive system. The reproductive systems themselves are pretty complex, and their life cycles are complex. So they produce lots and lots of eggs, lots of babies, because they have such complex life cycles that have a lot of risk that they have to produce a lot of eggs. Yeah, okay. These guys live in freshwater environments as well as marine environments, though many are parasitic. Okay, so here you have a little Planaria. Oh, I hope you can see this one. Oh, he's a little cut off. I'm sorry about that. So my pictures are a tidbit cut off. Let me see if I can fix that for you. Yeah, no. Ah, fiddle. Well, okay. Let me see if we can fix it this way. Maybe. Aha! Now you can see it. Okay, sorry about that. See, I had to check my window. Make sure you can see it. All right, so here we have a Planaria, a free-living flatworm. We're going to talk about each of these groups in turn. Here you have flukes, and here you have a lovely tapeworm. Yeah. Ooh, fun thing. I was really sad I didn't have my phone with me. I got my coffee, hold on. I was super sad I didn't have my phone with me because this weekend on Saturday when I was out in the field, I sample rocks to get the algae off of them, and then I take them back because they're part of the habitat. But I always check the rocks for organisms because I'm not trying to kill the organisms. I'm not sampling those. So I always, wipe them off and let them go about their business. And I don't want to kill them. I just want to make them late for something, right? So anyway, one of the rocks had a little planaria on it, and I was like, oh, I wish I had my phone because I could show you guys. They're so cute in the field. So I see these all the time in the freshwater environments that I work in. All right, so just an overview here, and I told you I'm going to tell you, and we're going to go into these later, but so the Lophotrochozoans, so these are the Platyzoans who we're going to focus on today, but just to kind of introduce you to the Lophotrochozoans, okay, they get their name, it means crest or wheel animal, but it's all based on two things. First, the name of the larva is called the trochophore. Trocho. Okay? So this is the trochophore larva, and they can be either motile or not, move a lot or not. Okay? And the Lophotrochozoans, so right now we're going to talk about the Platyhelminthes and the Platyzoans. Later we're going to talk about the Lophotrochozoans, which are protostomes that fall into the phyla Mollusca and Annelida, which you're going to be looking at in lab this week. Yay! Okay, so the first part of that name is the trochophore, and this is the big characteristic you need to know of these animals. So they have a trochophore larva, okay, that's got lots of little cilia, and this is how, if they are the motile ones, they will move around, okay, it's just with the cilia. And they have a feeding structure called a lophophore, okay. So in certain groups, so the Mollusca and Annelida have the trochophore larva, and then the Bryozoa and Brachiopods have a feeding structure called a lophophore in their larvae. And their larvae are sessile, okay, and these are the tentacles around the lophophore, but the lophophore is the feeding structure associated with these animals. And so they're called Lophotrochozoans because it speaks to, it describes essentially either larval stage or a structure in the animals of each of the phyla that are found within this group. So just to review, as we go back, the trochophore larva, okay, you have, this is part of Trochozoans, okay, and the lophophore is a feeding structure, and that's where they get their name, okay. And the four phyla within this group are Mollusca, Annelida, Brachiopoda, and Bryozoa, okay, which we talked about last time. All right. So, aside from those groups, and like I said, this is an introduction. This is introductory in chapter 34. Your book kind of gives you an overview, so I'm giving you an overview. So the Ecdysozoa, this is a damselfly molting. It's beautiful, don't you think? So these organisms get their name because they molt, and that's the name of the process of molting it. But Ecdysozoa means molting animals, and ecdysis is the process of molting. That's the scientific name for it. So you can sound really smart if you want to use that word. All right. So here's the thing. Within the Ecdysozoa, and I have blanks so you can test yourself, okay? Because we've already talked a little bit about these. So within these Ecdysozoa, this is two large groups. So one of them contains more species than any other phylum. What is it? Yes, very good. It's the arthropods. Exactly. And within the arthropods, you have a powerhouse of genetic and molecular biology, and that is Drosophila melanogaster, which is the fruit fly, right here. Okay, the Ecdysozoa have been very good to biologists because so Drosophila are a big powerhouse in biology, as well as, and I mentioned and talked about before, Cianorhabditis elegans, which was the first organism I ever studied. Right here, I called them all George, even though they aren't all. Well, I just called them all George. Okay. You have males, and then you have hermaphroditic animals that have both male and female sex organs. Um, but these guys are everywhere. Okay. And what group is that? What phylum is that? Nematodes. That's right. It's the nematodes. Okay. They're nematodes. So those are the two big groups in the Ecdysozoa, but you do have more. Okay, we're going to talk about the arthropods and the nematodes. However, you see these groups, so those are the main ones we're going to focus on, but it does consist of several other groups, including the tardigrades. Okay. All right. So there was one more thing I wanted to tell you, if I can remember it. Oh yeah. Your book says something that's really cool. I was just kind of looking through it earlier, and it says, I told you that nematodes are everywhere. You can find nematodes all over the place. Well, nematodes, if you were to take out, like in soil, if you were to just take off the soil and just like, it disappeared, but the nematodes didn't disappear, you would still be able to see the structure of what all was in there, because nematodes are so numerous. So, pretty cool. Okay, so let's get into our flatworm friends. Okay, all right. So this is phylum Platyhelminthes, and so what's really nice about this last part of the semester is that lab and lecture overlap quite a bit. Okay, so your lecture. Sorry, I get distracted by my dog. Can I see my dog again? Here. Hey, Vicky, say hi to everybody. Say hi, everybody. Hi. Okay, a little bit. Oh, you want to see him again? Hey, Zee. How you doing, buddy? I know, good girl. Okay, all right. And at the end of this video, I'll show you Morty. So stay tuned. Okay, he's recovered, I think, pretty well. So, anyway, the Platyhelminthes, the flatworms, these consist of 55,000 species. They're very successful. Okay. Here you have, we have represented the main groups. So the Turbellaria, the Trematodes, and the Cestodes. And we're going to talk about each one of these. Okay. So here, let's talk about characteristics of flatworms. So here you have a nice little planaria. Okay. So here, this little dude, these guys are acoelomates. All right. And let us go through the various parts of this, and I'm hoping you can still see it from mine. So you still see it if I do that? I don't know. Y'all let me know, okay? But you can look through the slides with me, okay? If it doesn't work, because I'm having to change the size of the window, okay? So here, these are acoelomate animals, all right? They have eye spots. Well, okay. Let me take that back. The turbellarians have eye spots. The free-living nematodes do. So here's the external anatomy. Okay. So this little dude, who's genus Dugesia, has eye spots. They can detect light. That's about it. You alright? My dog just gave me some attitude. And they tend to move away from bright light. Okay. And here is the mouth. And this is the pharynx. So this actually fits into their body. It kind of goes, well, it goes along their body, and then when they get ready to eat, it comes out, and it's kind of like a vacuum cleaner. In the lab, usually I will show folks, we'll have either like a little piece of liver or some dog food on a slide, and the worms will actually climb upside down on the slide, and we do it that way so you can see their ventral side, their tummy side, and their little vacuum cleaners will come out to eat. It's pretty cute. All right, so here, remember these guys are acoelomates. They do not have a coelom. Hi, what's up, Ziggy? Why are you crying? We'll go wake him up. All right, so internal anatomy. Here you have your intestine and your epidermis, right? Cool. So, and your acoelomate animal within it's, but this is mesodermally derived tissue here, right? Ectodermally derived tissue and endodermally derived tissue. All right. They have a nerve cord. Their nerves are kind of, they look like a lattice kind of configuration all up and down their body. Okay. You have, these are hermaphroditic animals, so you have both male and female reproductive organs. So you have testis on the top. So this is if you're, this is a cross section, right? So testis, sperm duct, oviduct. All right. So male reproductive organs, female reproductive organs. Okay. And they have, and then you have the musculature. You don't have to know, for me at least, you don't have to know the names of those various muscles or the musculature. I'm just showing you the musculature. But do be familiar with the reproductive parts. Okay. All right. So, as I said before, these guys, they they have cutaneous respiration. Oh, son. Ah, ah, oh, no. No, I'm trying to teach, son. Go away. It's okay. All right. Sorry. It's just really in my face. So, cutaneous respiration. So you have gas exchange right across the skin surface here. Yeast. Okay. And they have a really wide range of sizes. So here you have a beautiful marine flatworm. I posted on Schoology a nice video of some marine flatworms penis fencing. I like to think these guys kind of look like magic carpets. Aren't they cute? Okay. So huge range in sizes. From itty bitty, like about a millimeter, that's typically like Planaria, those little free living flatworms. They're very small. Like they could be super small. Okay. Up to many meters. Like many. Like this flatworm. So flatworms can grow Ifs. So they get really huge. And those huge ones are typically the flatworms. Not, excuse me, not this, they're all flatworms. The tapeworms, whereas the smaller ones tend to be like those free-living flatworms, and also flukes. Flukes are pretty small too. They can be. Okay, the trematodes. All right, so let us talk a little bit about their physiology. So depending on the organism within the flatworms, they can be carnivorous, or they could be scavengers if they're free living. Many of them, though, are parasitic. All right. There is only one opening to the digestive cavity. All right. And they do have extracellular digestion. And essentially, in the pharynx, so remember it's that little, in the free living ones, they have the little vacuum cleaner. You can't see my whole arm. The vacuum cleaner. This is the pharynx, and then at the end it's the mouth, right? That is attached here, all right? That food is taken in, and it's muscular contractions, but it kind of looks like what you would imagine like a cartoon vacuum would look like. Okay. All right. So they take in the food, and it begins to be digested. All right. Tapeworms. This is an important thing to remember. Tapeworms do not have a digestive cavity. They directly absorb food through the cell wall. So they do not have extracellular digestion. That's important to remember. Why don't you think tapeworms have extracellular digestion? They don't really need it, right? They get their nutrition directly from the host's blood, which we will talk about in a minute. So exciting. All right. So here you can actually see, this is again a little turbularian, a planaria. So here is the pharynx in the mid part of the body, okay? And it's kind of hard to see, but this is the pharyngeal chamber itself that goes into the gut, okay? So pharynx into the gut. Cool. That's really all you need to know about that, okay? They do have excretory and osmoregulation, osmoregulatory systems. And essentially, so they have their pharynx is like here and their intestines here, and here's the anterior part of the animal, okay? But they have basically little tubes called nephridiophores, which serve an excretory and osmoregulatory purpose, okay? And just to remind you, or if you don't know, osmoregulation is the regulation of solutes, or salts in the body. It's really important. It's super important. You can die if you do not properly osmoregulate. Okay. Okay. Talking about the nervous system. Again, I talked about this kind of lattice-like structure, a structure to the nervous system. Okay, so you have your nerve cords that come. These are the cerebral ganglia. That's essentially the little flatworm brain. And you have nerve cords that run down the body parallel, okay? And then you have nerves that go across, like a little ladder, all right? Very simple nervous system. And again, they have those eye spots that can tell them whether it's light or dark outside. Well, they're all outside, whether it's light or dark in the free-living flatworms. Now, most of these guys are hermaphroditic, again, male and female sex organs. So remember we looked at that cross section. You have your ovary and your testis here. So those are those gamete-producing organs. They are sexual in their reproduction, and that's where penis fencing comes in. Sorry, I love it's so interesting. Like you have to stab each. Ugh. Anyway, but these guys are really well known for their ability to regenerate. So asexual regeneration, basically that just means if you cut off their little bodies somehow, if you cut their bodies, it will regenerate into a whole new organism. So this is showing you that. Whoa! All right, so if you cut one, this is actually one where it had just been cut a little, it wasn't cut completely, and you have the beginnings of regeneration. So it will create a whole new organism. pretty crazy, right? Yeah, so you can cut one of these in half and you'll get 2 worms. We actually did that. We do that in the lab usually. We'll actually take one and or let a group take. Do I do it per group? I can't recall, but we actually will take flatworms and cut them up a little bit and then allow them to regenerate. And that sounds terrible now that I say it out loud, but you get more flatworms, so and they're fine. Though I mean, I guess they tell the other flatworms, horror stories like one time I was caught by aliens and cut in half, and that's how Jimmy got here. Anyway, okay. So within the groups, within the flatworms, you have 2 big groups. Okay, you have the Turbellaria, which that's a class. So kingdom, phylum, Platyhelminthes, class Turbellaria. Those are the free-living flatworms like Planaria. And then you have the parasitic flatworms, the NeoDermota, which they're named. It says new skin. Basically, this just has to do with how their skin, like their skin when they develop. You don't have to worry too much about that. But these are the NeoDermata, and that contains the trematodes, so class Trematota, and the Cercomeromorpha. So the Cercomeromorpha are the tapeworms, but the class of tapeworms is Schistosoma. Cercomeromorpha is a monophyletic group. It's a group. But Schistosoma is the class of tapeworms. So you're going to hear in labs cestodes, tapeworms, and I don't want you to get confused like, well, we're calling them Cercomeromorpha in the lecture. Well, this is a different grouping, okay? And I'm sorry about that. That seems a little confusing, but the Cercomeromorpha, so Schistosoma are just flatworms. Cercomeromorpha are flatworms and other related organisms, okay? Just a slightly larger group. So let's talk about each one of these, okay? All right, here we are with Dugesia. So Dugesia, this is what we often call Planaria. This is in class Turbellaria. This we don't think, oh, okay, let me just tell you about Turbellarian phylogeny. It's kind of a mess, and we're kind of still figuring it out. So this group is probably not monophyletic, and we're still figuring out the phylogeny of all of the free-living Turbellarians. So again, here's the anterior end of the animal, right? So you've got your little eye spots. Here's the muscular pharynx, and your intestine inside would be up here, right? The nerves go up and down the body, as does the reproductive system. Well, systems. System. They've got one system. It's just got male and female organs. All right, which is not uncommon at all in the animal kingdom. So, all right. So, got a couple videos I'm gonna let you watch, but please watch these. Actually, you know what? No, I can't show you the video, I don't think, on here. So please do watch these, okay? But we're going to move on from here. I'm finding that sometimes these lectures actually end up being shorter because I'm not stopping to show you a bunch of stuff. For better or for worse, we're kind of distilling a lot of the semester, but that's okay. All right, so within the NeoDermata, so we just talked about Turbellaria, the free-living flatworms. The NeoDermata are the parasitic flatworms. They can be ectoparasites, which live on the outside of their hosts, or endoparasites, which live on the inside of their hosts. Okay, these organisms, unlike the Turbellaria, don't have eye spots because they don't need to see. Trematodes and flatworms, but I keep calling them flatworms. Trematodes and tapeworms, which are flatworms, don't need to see. Okay, so they don't have any eye spots. And they have certain adaptations to fly under the radar of the host immune system. My Lord. Ugh. And to not be digested by the host. So they have special adaptations. And I'll talk a little bit more about this as we go on. But the main thing is, especially like with the Schistosomes, they use the host's own antibodies to fly under the radar of the host immune system. Tapeworms, most of them have some sort of cyst stage that has an outer cuticle or covering that protect the larva from the digestive juices of the host. All right, so within the neodermates, you have the trematodes, which are the flukes, and the circummeromorpha, which are the tapeworms and their friends. So remember, tapeworms are class Cystota, but the circummeromorpha group is tapeworms and related organisms. So some organisms that are not in class Cystota. All right. So let's talk about the trematodes first. There are quite a few of these. There are over 10,000 species, and they range pretty well in size. They can be really small, up to about 8 centimeters, which is like that. We actually have some in the lab. So when you get back to campus next year, if you want to see some trematodes, come find me. All right. They have mechanisms for attachment on the anterior end of the animal. So these guys have suckers or anchors or hooks, depending on the kind of trematode it is, the kind of fluke it is. All right. And many of them have life stages that require them to have 2 or more hosts. So they have an intermediate host or hosts, and then they have an ultimate host or hosts. And the intermediate host is where development happens. The ultimate host is where reproduction actually happens. Okay? And they produce, like I said before, lots and lots of eggs, lots of babies, because their life cycles, since they have to go from different hosts to different hosts, they're exposed to the environment more, which can be lethal depending on what the environment is like, right? They have a higher risk of dying. So since they have this really risky life cycle, they produce lots and lots of young. Okay, they're very fecund, is what we would say. Fecundity is how many young you produce. It's just, oh my gosh, guys, it's like, oh my gosh. Okay, let me do this. Let me finish. I feel like I'm getting sunburned right now. All right, you know what? We're gonna move. You win, son. You win. Let's go in here and talk about flukes. Never thought I'd say that. Let's just go in here and talk about flukes. We'll do the Morty reveal. I think I'm gonna have time. All right. So come with me into here to talk about flukes. Oh, you know what? Oh, don't you do it. Don't you do it. No! Okay. Sorry. But I don't want to stop the video. We're already so far into it. Okay. So, let's finish up with flukes. Z, would you like to help us learn about flukes? What do you want to know about? So, let's talk about flukes. Here you have, and I'm going to show you some life cycles of various kinds of flukes. Do you want to get up here and help me teach? Yeah? Okay. Go. Mad because I'm not throwing the ball. Listen, Ziggy, we have to talk about worms right now, okay? All right, so this is the life cycle of the oriental liver fluke, okay, Planorchus. So you have multiple stages, and a lot of these flatworms, their intermediate host is actually like a snail, like a mollusk of some sort. So here you have and I also want to point out most of these issues, most of these parasitic organisms like the trematodes or the cestodes, they occur and they're prevalent in places where you don't have very good sanitation because the way that they're transmitted is typically through fecal matter. So here you have an egg containing so a miracidium is the larval first stage of this liver fluke. Okay, so baby fluke in the water. So this is water that's contaminated with fecal matter that has these little eggs in there. And this is taken up and has to be taken up by a snail. Yeah, has to be a snail. So you have your snail. No, stay here. All right, who takes it up. And within the snail, the intermediate host, it will develop into further stages. So the miracidium is that first stage, and that develops into a sporocyst, which will in turn develop into a radia. And then finally, when it's ready to go on to the next host, it will develop into a cercaria that has a flagellum. Okay, so they can swim through the water, right? So fish, these get into the fish. The fish will eat the mollusk, all right? Or sometimes these are actually out in the water too. So depending, but they have a couple of ways they can get into fish, but they get into the fish, all right, and insist within the fish muscle tissue. Okay, so this is metacercarial cysts. So this is a cercaria, right? So this is the larval stage that insists within the muscle tissue of a fish, and then that fish is caught, and when that fish is caught, it is, well, you know what happens to fish when they're caught, right? They're cut up and eaten. Here's the thing. This particular liver fluke is passed on by eating raw or undercooked fish that are infected. Okay, so this is where you hear a lot about people saying like be careful with sushi and stuff like that. So this is the fish is eaten by a human, all right, where then that fluke will migrate particularly to the bile duct of the liver. That's why it's the liver fluke, okay, where it will reproduce and live out the rest of its life in peace and harmony. Not really. It's actually not good for you. Pretty severe liver fluke infection can lead to things like cirrhosis of the liver, which can be fatal. And yeah, definitely compromises liver function. Your liver is very important for detoxification. It is your detox. You know, people talk about detoxes. Listen, bruh, I got a liver and some kidneys. I'm detoxing all day. Okay, so that is the life cycle of the liver fluke. So what you need to know, I want you to know these life cycles because they're really important, especially if any of you ever want to do, you know, I have had students who are interested in doing like tropical medicine or Doctors Without Borders and stuff like that. You're going to see stuff like this. But you see that stuff like this even here, but just because of the fact that you can have imported fish or fish even from here that you eat and they may be undercooked, right? Or sushi. So here you see the actual organism. So you have a ventral sucker. That's how it attaches, all right, and the rest of its body, which you know all about its anatomy because you drew a picture and labeled it. Good job, guys. All right. So that is the liver fluke. There's one more thing I wanted to tell you about these. All right. Well, I guess that's all you need to know. Here we have, and here again. So here are the actual anatomy. All right, so you have your ventral sucker, right? The pharynx here. This is the intestine, right? The intestine goes both ways. It's parallel, all right? You can't really see the excretory tube very well in this actual organism, all right? So, sorry, this is the oral sucker. Duh. The ventral sucker is over here. You have multiple suckers on the animal, okay? So oral sucker. But here's the thing. Sometimes I've seen this referred to as the ventral sucker as well. I've seen both of them referred to as ventral suckers before. So that's not actually incorrect. But oral sucker, ventral sucker, okay? And then uterus, right? So this stuff here is the uterus, all right? And the seminal vesicles are a little harder to see within this, I will say. All right, so the male and the female reproductive organs are in here. The main thing is you have, so your uterus, all right, and your seminal receptacle here, okay? These are, this is your ovary. So ovary and seminal receptacle, all right? Here's your testis. One testis, one testis, two testis. Okay, so let me go back through this again. All right, so uterus, all right, up here is the gonopore, uterus, seminal receptacle, ovary. I hope I didn't call this a seminal vesicle. Seminal receptacle. The seminal vesicle is up here. All right, testis, testis. All right, and then it excretes out here. All right, cool. And then, oh, actually in here, I don't know if it's showing you very easily. They do have a bladder for excretion. It's just very difficult to see on here. Okay, so those are the main parts that I want you to know. All right, So I don't spend as much time, saying you have to know the, like the seminal vesicle is farther up, and it's kind of here. It's hard to see because of the uterus. Okay, all right. So let's talk about other kinds of flukes. So Monogenaya, this is actually a whole group of flukes that are ectoparasites. So they are found on the outside of animals, and particularly fish gills, you'll find these. But they can also be on the skin, all right? And their only ultimate host is like a vertebrate. And particularly, you'll see these in fish. So these are external flukes. That's the Monogenaya. All right, let's talk about blood flukes. So Schistostoma, here you have schistostoma. This is actually a schistostome. These are schistostomes mating, right? So the blood flukes, these are a big problem for humans. They cause schistosomiasis. Um, and they're especially a problem in tropical countries. So you have tropical Asia, Latin America, warm places. You have schistostoma, and it kills lots of people. And over 200 million people are infected with schistostoma at any given time. So, and it kills lots of people. So here's why, and here's what happens. So this is actually, so you have the male and the female, they lay into each other. So this is the smaller male, like, lays in a groove. Okay, so that's how they mate. And these guys, particularly they infect the blood cells, the red blood cells. That's why they're called blood flukes. And they cause lots of issues. And so this is actually a child who is, you see the distension, the distended belly, that's a hallmark of the infection. And it can damage your organs and lead to death if it's left untreated. But it causes fluid buildup in the belly. And so that's actually one of the hallmarks of the infection. So how do they get into you? Well, once again, snails. Man, snails. What is the deal with snails, right? So basically, when you have lack of sanitation. So in an area that has not a ton of great sanitation, right? You can have the snails that have the infectious worm inside them, all right? And the larvae of the worm actually can penetrate the skin of a human. So if the human is out, like for example, this is showing an example in a rice field, right? And the the snail with the worm inside is in the water. They migrate out of the snail and into the human. Okay, um, and cause lots of issues, lots and lots of issues in terms of organ damage. You know, particularly they cause issues for like the liver. I'm trying to think of the main other parts of the organism that they infect and cause issues for, but the liver is a big one, um, and well, so your excretory organs, kidneys, things like that, urinary bladder, okay, but they are a systemic issue. They can affect pretty much your body, okay, but those are the main ones: the liver, kidneys, things like that. All right, let's talk about the Cercomeromorpha. So these are the tapeworms. So we were just in the trematodes. Those are the flukes. So the cestodes and their friends. So these are the tapeworms. Here it is, the yearbook picture, 20 something about a tapeworm. Yearbook picture? It needs braces. No, I'm just kidding. So there are 12 species um, of tapeworm that specifically infect humans. And the main anatomy of a tapeworm consists of the scolex. They have a scolex and a neck, but sometimes the neck is thought of as part of the scolex, which is the top of the animal that consists of its hooks and suckers. So here's suckers. There are 4 suckers. There's one back there you can't see, and hook. And then proglottids, which are the segments of the worm that are each a complete reproductive system unto itself, and each one is capable of reproducing. Okay. Meow. So here, again, here's the Scolex. This is, the top with the neck, but this is the Scolex. We have a really beautiful model of a tapeworm Scolex that I bought for the lab. So I hope you guys will come by and ask to see some of this stuff next year. But it looks like this. So I mean, So here's the Scolex. Here it is on a slide. So you would look at slides, and here you have the sucker and the hooks. And this is how it attaches, particularly attaches to the host's intestinal wall. Okay, that's what happens. All right. Oh, if you've ever had flukes or a tapeworm, feel free to let me know. I would be so interested to hear your experience. I've never had any of these guys. Knock on wood, hopefully. So, though I have eaten a lot of sushi. Okay. Yeah, every time I teach this, I'm like, oh. But, so you have your Scolex and then your proglottid. So again, each proglottid has its own genital pore and uterus. These guys can reproduce. Each one of these can reproduce. And they just keep adding proglottids. That's how the tapeworm gets so long. So they attach to the intestinal wall with their hooks, right? And what happens is the proglottids, they will shed these proglottids over time, and those proglottids will come out in the animal's feces, and that's how the worm's life cycle continues. Okay, so here you have the anatomy of a particular proglottid, a mature proglottid, right? So they keep adding them, and they just get longer and longer and longer. Okay, and so each proglottid has a genital pore and a full suite of reproductive organs, because these organisms are also hermaphroditic. So you have, and it's hard to see here. So I'm not gonna show you a slide on here and go, hey, where's the testis? Where are each of the testes, right? Or where's the uterus? But do know that you have each proglottid contains a complete reproductive system. So this is basically a bunch of reproductive systems. Gulp. All right, so let's talk about a couple of these, and then we'll be done for today. So, is it today? Today. So Tonia. Tonia is a genus of it is a flatworm, a tapeworm that infects humans via the ingestion of rare beef. So you might notice, you might be like, wait a minute, what if I just didn't eat these animals? Well, then you're less likely to get it. You're still unlikely to get it. It's pretty rare, especially around here, if your animals that you're eating are coming from a place with good sanitation. Well, relatively good sanitation. Yeah, pretty good sanitation, but good sanitation. But you can still get them. I'm not telling you not to eat meat or to eat meat. I'm just saying that's the risk. So, particularly, or you can make sure that what you eat is very well cooked if you do eat an animal, because that way it kills these organisms. Okay, so let's start here. So here's the thing. It starts in the cow. All right. So the cow, and like I said, most of these are more prevalent in places with a lack of good sanitation, because most of this is spread fecally, fecal matter. So if you have an area that gets wet a lot, right, and somehow gets contaminated with sewage or has been, and then a little cowie goes out there and some of the grass has eggs on it, okay, then it can take up the worm, the larvae. Okay, so those larvae will insist within the muscle. Do you notice the intermediate host? Typically, the larvae will insist within the muscle. All right, and then so just like with the fish with the tremontoes, the flukes, that insisted those insisted larvae are then ingested by humans. Now, particularly rare beef, so beef that has not been properly or fully cooked. So like everything my husband eats. I'm sure he has a tapeworm. Well, no, he doesn't. I mean, He might. He may. Maybe someday. I don't know. No, I'm just kidding. Beef here is pretty good. You don't have to worry in the United States so much. Though it has happened. It's just really, really, really rare. Okay, so anyway, you eat that mussel, all right? And here's the thing: these worms have, like I said, a really a cuticle or a shell around them to keep them safe. All right. So once they get into the intestine, they can attach. They mature and attach, and then the proglottid production begins, and they just keep adding proglottids and from this end. Okay, so proglottids keep being added from this end. From this end, the proglottids are the proglottids that are um, reproducing actively. So gravid means, essentially gravid is a synonym almost. It's not the same as pregnancy, but being gravid means being ready to reproduce. All right, so on this end, you're adding new proglottids. At this end, you're shedding old proglottids that are gravid and ready to go. So you have basically this um, assembly line of reproduction. Okay, and that gets out again. That's shed in the feces of the human, and if you have an area with not so great sanitation, and that sewage gets out and can infect grass that the beef feed on. Okay. Another example of this is the broadfish tapeworm. So this broadfish tapeworm has a couple of different hosts, and this is really interesting actually because the broadfish tapeworm actually changes the behavior of one of its intermediate hosts. So the broadfish tapeworm, the first larval stage actually will is free swimming, and it looks for copepods. This is a freshwater. There are marine ones too, but here we're talking about freshwater. Freshwater, a freshwater crustacean called a copepod. They're very small, and they infect the copepod. And what they do is they basically make the copepod search for light. Well, when you do that, that's almost like with toxoplasma, another organism we've talked about. Remember, I told you toxoplasma makes mice seek out cats. So the second larval stage of the broadfish tapeworm causes copepods to look for light in the water column, essentially. So they're basically going higher up in the water column as opposed to trying to swim in places where they would be less likely to be seen by predators. And that is actually a life history strategy of the worm, where they can be taken up by the fish, which is the next intermediate host. See, wow, it changes the behavior of this first intermediate host. They're taken up by the fish. And then the fish, its ultimate hosts, are mammals. So not just humans, but also any mammal. So this broadfish tapeworm can infect mammals, humans, as well as bear or other mammals that may eat the fish. Um, and the bear, they are much more likely to end up with the tapeworm because I I mean I have not seen a bear frying up fish. If you do, call me. All right, so they're eating it raw. So most of these, the issue is um, undercooked meat. Okay, so okay, you know, you don't have to worry so much about like the meat itself, except just make sure you cook it really well, right? All right. Also, just and we're done. I'm sorry I've gone over a little bit, but this is showing you a human brain with dog tapeworm cysts in it. That's what these are. So sometimes even in the right host, sometimes these organisms will invade parts of the body that are not its normal tissue tropism, its normal tissue preference. Tissue tropism is showing preference for a tissue, going to a particular tissue, like liver flukes, they go to the liver, the bile duct of the liver. So these hydatid cysts, you especially see this though in when you have a parasite in the wrong host. They either if the parasite survives, it can wreak havoc on that host. So again, this is a dog tapeworm, but the tapeworm went to that brain. So that that's the lecture today. Oh man, what a way to end, right? Um, I'm already way over, so I guess I'm just gonna. You know what? If you want to see, okay, hold on. I wonder if I can show you my whole self right now. Can it do it? Yes. All right. Let me show you Morty. Come in here with me. Come with me. You'll be in a world of pure investigation. I don't know what to say. All right. Let me get the. Here, let's fix this. Oh, sorry, guys. Please just let us go. You can turn off the video if you want. I'm not. Nobody's told you. We're done with lecture. Nobody's putting Morty to your head. Okay. Are you ready? Dun, dun, dun. Yeah, he did. He's stuck. Don't you love? Yeah. All right. Okay. So that's all for Monday. And we do have a lecture for Wednesday, but no lecture. You don't have to watch a lecture on Friday. Okay. But it's technically Easter break. So sometime this week, take your test. Make sure that you watched this or read the Schoology update for this week. And yeah. And we're still going to have a Friday Q&A just in case you need me, but it is absolutely optional, and it's even extra optional this week because it's Easter break. It's Good Friday on Friday. So, okay, let me know if you have questions. Put them in the discussion board, or, yeah, email me. All right, have a good day.